Good afternoon and welcome members. Amanda, if you could help with continuing to admit people as they arrive in the waiting room, I'm going to go ahead and get us started. I'm Ann Erkowich, Member Engagement Manager, and we're so grateful to all of you for your support. And as a member, you're a change maker. You support a wide breadth of work in your region to fight climate change, save endangered lands, restore ecosystems, and to make sure that the waters of Long Island Sound is healthy for all of us. This afternoon, Elena Colón, our fantastic laboratory manager, will share a behind-the-scenes look at how Save the Sound's impressive lab team and its John and Daria Berry Foundation Water Quality Lab contribute to preserving and improving water in the Long Island Sound. There'll be time for questions at the end. If you think of questions as you go along, or as we go along, feel free to put them in the chat and we'll get to them at the end, or you can save them at the end and unmute yourselves and we can chat. All right, and with that, I'll hand it over to Elena to get started. Thanks.
Hi, everyone. I'm Elena. Thank you for coming to this virtual lab tour. Hopefully I can make it feel like you're in the lab as much as possible. I think I have a lot of great videos and photos of our lab. So I am going to go over what we have here. So just an overview. I would like to introduce the laboratory. So myself, I like to tell you a little bit more about my background, my professional background, and then of course, one of our other full-time staff who we hired last year, who's really important and couldn't do it without her. And then I cannot tell you about, our lab is relatively new. And so I think the story in our journey to building this new lab is really impactful. So I want to show that. And I think once you see, our growth, that I'm amazed. So hopefully you guys are too. And then of course, I'm going to show you our new lab, and I'm going to go through the different types of analyses that we run in this lab, and where we're going to go next with future analyses. And then of course, I'm going to give you some information on how you can get involved if you want to take action for cleaner water, and some volunteer opportunities. And then we can take some questions. And also I have a little photo of here of two of our interns that helped out last year. They were amazing, Heather and Scarlett, and they rocked. And we'll get some new ones this year. All right, myself. So my journey into the environmental sciences started back in Philadelphia. So back in 2013, I was hired on as the diatom lab supervisor at the Academy of Natural Sciences. So I don't know if anyone has ever attended the museum in Philadelphia. It's part of Drexel University, and they have a really well-established environmental research center with many labs and capabilities. Besides, I don't know if anyone knows much about diatoms. I put a little picture there, and they're unicellular algae, microalgae, and they're really pretty. There's an estimated probably like 12 to 30,000 species. I mean, the exact number isn't known, but they do produce about 20 to 30% of the oxygen that we breathe. They're really important, and they're a really good indicator of water quality. Not an easy algae to identify down to the species, because there's so many and they look so all alike, but they're really good indicators of how the water is doing. So that's the work I did. And I also got heavily involved in a lot of field work. So I did a lot of freshwater mussel surveys, a lot of snorkeling in the Schuylkill River. I don't know if you know much about it. Definitely needed to shower after that. And the Delaware River Watershed Initiative was a huge, and it was like a $35 million funded project. And, we did all types of monitoring work in the Delaware River watershed to characterize it based on macroinvertebrates, chemistry, water chemistry, algae, habitat assessments, fish. So I was very much involved in helping collect all of that information. So and then I moved. I mean, I was, sad to leave there. I loved my time at the Patrick Center for Environmental Research. But, I moved to Connecticut, and I luckily joined Save the Sound in 2017. So I've been with Save the Sound for a while. And originally, I was brought on as a seasonal water quality coordinator, but they kept me on. And that's kind of like my thing. I did the same thing at the Patrick Center, just kind of stick around until they take me full time. That's my tactic. It's worked for me pretty well for me so far. And then, a lot of growth in the programs. I came in at a really great time. And then I was hired on or as a full-time environmental analyst. And then in 2022, there was a transition. Now that we had this brand spanking new lab, there was a lot more work to be done. I wanted to kind of come full circle and get back in the lab. I love the field work, but I was ready to move on to the next challenge. And so that's what I'm doing now. Very excited. And then I want to introduce Lindsay Potts. She's our full-time laboratory technician. She comes with a really extensive chemistry background, which is what we really needed, since that's something that we added to our lab is inorganic chemistry analyses. And so she attended UConn Avery Point, and she received her master's at McGill University in Montreal. And so, yeah, she worked in the marine stable isotope lab. And then she also managed the lab at the forensic science department, but she wanted to come back to the environmental sciences, and she wanted to get more hands-on. And so we were happy to have her, and she joined us in July of 2023. Yeah, she's very integral into moving things forward. And once I show you the number of samples that we have in the lab, you'll definitely agree with that, we needed extra hands. So this is the old lab. So I kind of did a little photo collage, and it's going to kind of loop. By the way, that's our bathroom. That was our only source of sink and water. This is literally our conference room, our lab, field storage room, my desk. This is what we had. We made it work. I was constantly reorganizing. I had stacks of coolers next to me. We made it work though. And, I'm really happy, with what we made of this. But yeah, we definitely, you can see for yourself, we outgrew it. By the way, you see those red bags in the back. Please remember that, because I'm going to talk about that a little bit. Those red bags that you saw, I'm going to tell you what those are. By the way, that was our only closet as well. So I was constantly rearranging that closet. But hey, we were successful nonetheless. So yeah, lots of equipment, and I'll tell you a little bit about the equipment. It's another aspect. It's not necessarily having to do anything with the laboratory directly, but we do have this large equipment loan program for one of our water quality programs, and I manage that as well. So that was from 2014 to 2021. And then, so before I show you the new lab, our current laboratory, I want to talk about the programs that we have. By the way, this is not to scale, but hopefully it gives you a good visual of numbers. And of course, you'll see 2019 in yellow, because that's kind of the year where things kind of blew up, and that's when we're like, okay, we need to start thinking of the future, where we're of our space. So this is bacteria samples. So in 2014, we collected 209 samples. This is back when Tracy Brown, who is now Riverkeeper, but she was our wonderful regional director, and she started this program. And this is before we even had the equipment, the lab equipment necessary to actually analyze bacteria samples. So they collected it, and then they took it to the Westchester County Environmental Laboratory. And then in 2015, Peter Linderoth, who's our director, came on, and he was able to acquire the lab equipment through an EPA equipment loan program. So he got like the necessary lab equipment, and he has a scientific background to be able to really do the analyses in addition to collecting. So you can see we just, tripled by 2017. We were able to triple the number of samples that we were analyzing. And that was because also we lowered costs. We were no longer having to take it to another laboratory. We were able to just only have to worry about buying the consumables, which were in bulk. So this lowered the cost significantly. And we just kept on growing over the years because of the success of the bacteria program, which, by the way, on our website, we do publish our results, especially during the summer season on a weekly basis. There's the Sound Health Explorer. We will share all of those links for you, for those who are interested in checking it out. I highly recommend. And then as we, continue to grow, we were also able to, purchase, our own lab equipment, and the more lab equipment we were able to do, the more type of analyses. So we're just growing. And I also want to point out, I think starting probably with the 2021, 2023 numbers, if you go on our website and you see, oh, we collected this, these numbers are higher because I'm also including what we call exploratory samples that we ran. So throughout the year, because now we were not, relying on a loan program where we only had the equipment for, a certain number of months, we had, lab equipment all year round. So if we got a report of, maybe potential sewage pollution, sewage getting into the water, we can go grab a sample. And I have done that in the middle of winter, anything suspicious. So we're able to continue that. So again, that's part of the growth of the number of samples. And also you'll see later, but we were able to, duplicate the number of equipment. So that means we can run more types of analyses of a bacteria. And again, that's why it increased. So in 2017, that's the year I came on. And that was the first year we really started the unified water study. And again, we talk about that on our website, and throughout the presentation, I will continue to bring up the unified water study. It's an ecological assessment on the Long Island Sound bays and harbors. We realize, hey, there's a lot of data out there, and the deeper waters of the Long Island Sound, it's collected by New York and Interstate Environmental Commission and Connecticut DEP. But these bays and harbors, which are really impacted by very localized conditions, it's shallower waters, did not have as much data. So we're like, okay, let's do this. Let's get let's partner with other existing organizations and monitor all the same way. And so we are the managing organization. So I won't get too much into that because I know I can certainly answer your questions if you're curious about that. But we started off with 12 in 2017, and then we almost doubled by 2019. It was like, whoa, like it really just kind of took off. And, if you saw in the old pictures, all of that equipment, we loan the equipment to the groups because it's very expensive. One of the key pieces of equipment costs, about almost $17K. So it's not always affordable for just a group to random, like, okay, yeah, I'm going to just buy this. So, and I have more technical, experience with the equipment, so I can really train them on how to use it and also do the repairs and maintenance. So 2019, it just kind of blew up, and we're constantly adding groups, like every year. So we're at about 26, 27 now. So next is chlorophyll samples. As of now, the Unified Water Study is the program that produces all of our chlorophyll samples. And so, of course, since the groups increase, so do the number of samples and that we analyze. prior to us getting set up in our new current laboratory, we had to pay, contract a laboratory to get that those analyses completed. And I'll tell you why later, we really needed to move in the direction of where we were doing it ourselves. And then, we kind of dipped down a little bit in, 2021, the pandemic. I don't think a lot of people could make as many trips out. So that decreased the number of samples, but we bounced back. And then nutrient samples. I mean, oh my goodness, there's so many samples. We have 5 freezers, tall freezers, just packed, and you're gonna see that later. So in 2019, we started to conduct the tier 2 aspect of the unified water study, and that entailed collecting nutrient samples. And so that has increased significantly. And that was another thing that we realized we really need to be able to analyze these samples in-house. I'm only getting data back just recently, a few months ago, from like 2019. So we've had to wait a long time in order for our contract laboratories, number one, to be able to handle the volume. And then, of course, they have other projects and clients, that they're processing samples for. But, now that we're going to process them ourselves, we are our primary customer, and, we don't have to wait as long. So just want to go back to 2019 real quick. That's when we started having conversations like, okay, we need to move. And then we actually started, going around and looking at different spaces. And then the pandemic hit, but we just kept on forging on. And it was during that period where we were able to build it out, find a place, have an architect, draw it out. We were very heavily involved, especially the laboratory. And then end of December 2021 was kind of when it was completed. And then we moved in 2022 after we came back from the holidays. So the John and Daria Berry Foundation Water Quality Laboratory. So this is what it looks like for those who haven't had a chance to visit. And this is how this video that I took, this is how it looks like. I didn't clean it up or anything. So it's just, this is how the lab looks. And you can see all of our freezers and our refrigerators, all of our coolers. And I'm in there. I go crazy with the label maker. I like things organized. I'm constantly rearranging where I put things because, there are future instruments that we plan on getting. But yeah, I'm pretty happy with this lab. It makes me happy. And so I'm going to move on now. I want to talk about what type of analyses we currently conduct in the laboratory. And so that would be pathogen indicator bacteria. It's often interchanged with like fecal indicator bacteria. And then we have nutrients. I'll get into that more detail about each type of analyses. And then we have chlorophyll A, and then total suspended solids and harmful algal blooms. And then I'll also get into things that are gonna be next is on the horizons. So the first type of analyses that, and this goes all the way back to 2014, pathogen indicator bacteria. And again, myself and others will say fecal indicator bacteria. I feel like saying fecal is like more impactful. People kind of like immediately get it because pathogens, are disease causing viruses and bacteria. But the fecal indicator. So the types of bacteria that you see here, Enterococci, E. coli, fecal and total coliforms, those bacteria specifically live in the intestinal flora of warm-blooded animals. So that's like geese, that's dogs, that's us, humans, we're animals. And so of course, intestines, fecal contamination is what we're measuring in the water. And sources of that, could be sewage spills, untreated wastewater, sewage overflows, contaminate, runoff, stormwater runoff, and of course, animals too, they contribute. And I wanted to highlight before we get in, I have a, pretty cool video to show you the actual method, but you probably think like, well, why aren't you just testing for the actual pathogens? Like, because enterococci specifically wouldn't necessarily get you sick. Testing for pathogens directly can get really complicated and it gets very expensive. There's not an easy existing methodology to do that. So it just makes it not practical to do. So, a lot of studies have already been established and done where, testing for enterococci and E. coli, is a very great accurate way of testing for fecal contamination. So likelihood of, if you have really elevated levels of these bacteria in the water, then yeah, there's probably going to be some dangerous pathogens in there that, so you don't want to interact with the water. So something's going on with that. So let me here. So this is a video. This is just, we compiled a few videos of some of us, setting up. So we use what's called like an IDEX method. So these are quantitrays. And then you'll see in these little, where it says Hardy, that's our dilution vials with DI water in there. And we like to do a 10 milliliter, dilution. So we're taking 10 milliliters of the actual sample collected from rivers, from shorelines, from beaches, which by the way, we collect from like about over 60 stations every week between June and end of August. And then you can see here, you see that powder there? I'm gonna, pause that a little bit. And you can see that power. I like to call it bacteria food. But what it actually is a type of nutrient indicator. So it's a reagent. It reacts specifically with the target bacteria that we are analyzing for. So that means like any other bacteria that's in there is not going to get metabolized by this indicator. And then I'll show you, it'll go into an incubator and it causes that bacteria to fluoresce. So I'll get into that a little bit, but yeah, that's why I call it bacteria food. So there's specific reagents that we use. So for enterococci, we use something called EnteroAlert. And then when we want to look at E. coli or total coliforms or fecal coliforms, we're going to use something called ColAlert 18. Oops, did not. Let's play this again. Pipetting is something that, I always go over, I'm like, are you at eye level? I'm actually, I don't know if anyone has seen me in person. I'm a pretty short person. So it's great for me. Peter Linderoth, our director, he's pretty tall. So he has to like really hunch over just to get to the meniscus of the, the pipe when he pipettes. So I feel like we try to compete sometimes, I think I'm faster at pipetting. So we then, after mixing it, the dilution and putting the food in there, that goes into these trays and they have like a certain number of wells, and then we seal them. And you can see we have 3 incubators, and that allows us to analyze, at one time, 3 different types of indicator bacteria. So we have enterococci, E. coli, and fecal coliforms. And there is a reason why you can't just put all of them in one incubator. Enterococci, for example, needs to, be incubated at a specific temperature for at least 24 hours. E. coli is at a completely different temperature, about 5 degrees or so less. I try to put it on there on the incubator so everyone remembers. And then it's for 18 to 22 hours. So there's a reason for that, why we need more than one incubator. Right here, after, you can see that this is what it does. So that bacteria food that I said that, gets metabolized by the target bacteria, it incubates at a specific temperature for a specific length of time. We pull it out and then it's going to fluoresce. So any bacteria that's in there, the wells will fluoresce. And so we check it off with a black marker. And that there's a specific, what we call a mean probable number, which is equal to a colony forming unit. That's a unit of measurement to determine, to quantify bacteria. That'll correlate. So there's like these little tiny wells, if you can see that, and then there's these large wells. And if I go back, I think you'll see there's like a, let me see if I can pull it up. So I try to show the table right there. So this table, will give you the number, the levels of bacteria that's in there, by crossing the small wells with the large wells. And this is a common method that, anyone that says that they analyze for, indicator bacteria, they're probably using this method. Oh yeah. So remember those red bags that I said, hey, remember, like in that little closet next to our bathroom where our water purification system. Yeah. So those are biohazard bags full of these trays. Do you remember the number of samples? Like last year we did over like 800 bacteria samples. So that was like 800 and something trays. And that's not even counting, lab blanks and duplicates that we run. So they need to be autoclaved. So an autoclave sterilizes things, instruments, etc. Well, we didn't have an autoclave back at our old office, and there was a lovely person at SUNY Purchase who, because she worked in a lab, and she would kindly autoclave some of our trays, but I couldn't just dump them all on her at once. So I had to, deliver one bag here and there. So we just accumulated all of these bags of like 600-700 trays that because you can't just dump them in the trash. You need to sterilize them, first. I mean, yeah, I won't be caught throwing out biohazard bags unsterilized. So yeah, it's so great to have this autoclave now, and be able to do that work. Which, by the way, the autoclave is used for other things, but this was an important thing for us to be able to do. So I just wanted to really point that out. So yeah, and we keep this in our freezer storage room. So. So now I'm going to move on to chlorophyll A, and chlorophyll is the predominant type of chlorophyll found in, green plants and algae. And it can be a sign of degraded water quality if there's an increase of algae. So that's something that we want to really watch for. So again, the unified water study, these are one of the key parameters that we measure, and it can really impact levels of oxygen. And that can happen if you have too much algae, then it's going to throw off the balance. Okay. And then, eventually it's going to decay. And then at nighttime, it does a biological process of respiration. So it's going to really remove that oxygen from the waters. It's going to just plummet. So, it naturally goes up and down, but you really don't want to go below, oxygen levels that are like not supportive of aquatic life. That's like when you see a lot of like fish kills, which is like terrible. And I've seen them when I've been out in the field. So that's something that we measure to see, okay, if there's too much algae, that's concerning. And, the biggest contributors to, huge amounts of algae would be really high levels of nutrients due to, again, sewage. You're going to see a running theme: sewage, untreated sewage, runoff, etc. Nutrients being, I'm going to get into nutrients as well, so I won't get into it, explain it too much now, but that's going to be kind of the running theme. And so I have another video. And so, is that like, did I not mute this? So you can see here, we get chlorophyll samples. There's a 28-day holding time. So we have these overnighted to us from all of the groups, in a cooler, and we process it. So this is just kind of showing you how it's run. These are actually old samples that, they were just like backup samples that we had collected. And so you'll see here, they get the filter itself, they get filtered out in the field. And then they, the actual chlorophyll gets placed, it ends up on this filter. It's a glass fiber filter. and then it goes into the tubes, and we add acetone to it. So it extracts the chlorophyll from the filters. And then we put it in a freezer, which you'll see she's going through multiple samples. I would say within a batch, we're processing, around, I think around 18 or so. It can be pretty time consuming. And I just want to say, usually when we actually are processing like the real samples that with the data matters, those tweezers, we do acetone rinse between, because you don't want to cross contaminate. So we actually have these, just FYI, in case there's anyone on here like, oh my goodness, like you're not being very clean. Let me take this, but we are, don't worry. And we put so we're putting acetone in there, and each one, they get capped. You see there, this is a dispense set, so it's already set to measure exactly like 5 milliliters of acetone. And then and then it goes into this box. So when we do process our chlorophyll samples, we want to make sure that it's not exposed to any significant amount of light, and that it's stored in this box. She's showing it off. I'm pretty proud of this, because I was told, many use like a cardboard box, wrapped in like duct tape to kind of like keep it hidden. And I was like, oh, something nicer and neater. So I got these little black bins. I got them on Amazon, and they're like these toilet paper rolls that you can store them. And then I bought these little like handles and drill them into the sides. So I made a few of these. So I got really excited about that. So you'll be in the lab, you're often, they do have existing, equipment that you could use, but sometimes, you gotta be a little creative to, make something that kind of works for your own needs. So I was excited. I really put a lot of work in it. But yeah, so yeah, she's showing it off. So it's gonna go into the freezer, and so we put it in there overnight. So we really want to allow that extraction process to happen, for at least like 18-20 hours or so. And you can see here, usually we would remove the filter from there, and then we have to centrifuge it before we go ahead and analyze it. And you can see here, she's putting 4 in there, and then like everything's preset. So that was like, important piece of equipment. This is what reads the concentration of the chlorophyll. This is a fluorometer. And so how it reads chlorophyll is, a certain wavelength of light is going to be emitted. Okay. And we have to clean the bottom of the tube, and basically the chlorophyll electrons, absorb that light, and then they kind of jump up, and then they fall back down. And when they do, that energy is released as fluorescence, and you can see measure fluorescence raw. And that's how we get that concentration. That's how that technology works. And then specifically, we do what's called an acidification method, because in estuarine waters, there might be an interference of another type of like chlorophyll, which is phaeophytin. It's basically called like dead chlorophyll, but it also has the same pigment. So you wouldn't be able to differentiate from the chlorophyll and phaeophytin that's in there. So we do an initial reading, add hydrochloric acid, centrifuge again, and then put it back in. And basically the acid kills the chlorophyll, so it no longer will fluoresce. Only the phaeophytin in there will contain, doesn't, is not, impacted by or affected by the acid. So then you'll do like a little equation and calculation, and then I'll get you the final chlorophyll results. So just adding a little science in there for anyone interested. And this is actually one of our low standards that we read. Okay. So now moving on to nutrients, and specifically you know, we look at nitrogen and phosphorus. So when you often hear nutrients, and we say nutrients because nitrogen and phosphorus is, you know, food for plant life, algae. You know, if anyone buys, you know, a bag of fertilizer for, you know, I have some for my house plants, you'll see like the major components would be nitrogen and phosphorus. And so there are different sources of nutrients. And again, you know, when I say there's a running theme, you know, a big contributor, you know, is usually untreated wastewater or runoff that ends up. And then you can see a connection, you know, of course it impacts, you know, fecal bacteria, it elevates that. You have chlorophyll, you know, blows that up. So we're measuring all of, you know, all of these type of analytes to assess water quality. And the nutrients that we receive are a result of the Unified Water Study right now. But there are other projects in the works that, you know, we will be processing nutrient samples for other projects. And so specifically ammonia and nitrate, those are that falls under nitrogen. They're nitrogen species, dissolved nitrogen. And then you have orthophosphate, again, is a type of, you know, phosphorus. And so these are the type of analyses that we run. And we really would have not been able to do this in our old lab. I mean, we need a fume hood. I'll show you soon. We really needed the discrete analyzer, which is a pretty important and technical equipment. So here, this is just, you know, a few of our samples. I think it took probably a month or two for Lindsay to label every single bottle. You know, we need to have a unique ID for every single one. And there are 60 ml bottles here kept frozen until they're ready to get thawed for analyses. We have the blue and the green tape. So the blue is to kind of like, it's just a visual, you know, even though the sample ID will indicate if it's unfiltered or not, the blue is filtered. So that's what you use to look at things like ammonia, nitrate. Those are considered dissolved nutrients, an orthophosphate. And then the green is unfiltered, total nitrogen, total phosphorus. In order to analyze for nutrients, we have to make so many standards and so many buffers and reagents. I think Lindsay probably spends the majority of her time mixing these. You know, some of them expire within a day. You have to make it fresh the day of the analyses. So, you know, this is just giving a peek of, you know, her making a solution. Another thing that we acquired, you know, with the new lab, besides the analyzer, was the analytical balance. It's definitely an expensive piece of equipment, but it's absolutely necessary. You can see here, we had to buy tons of volumetric flasks. You know, she's here mixing things, a fume hood. Again, we're dealing with some pretty, you know, abrasive chemicals, you know, acids, flammables. So we really needed, you know, a fume hood to meet safety standards. And then here, this is our actual discrete analyzer. This is an auto sampler. And you can see here, those wedges that she pulled out is where we pour the reagents and standards. I'll get into the science behind that in a little bit on why we need all these reagents and standards. And she's labeling. We keep very close track. You know, as you can see, this is not something that you can just kind of like throw in there. Like, you know, we need to always be very detailed oriented and meticulous. I have tons of tables and reference materials that we keep close by to keep track of, like, they're all numbered, you know, which reagents go in which, because the last thing you want is, you know, actually put the wedge in the wrong slot. So that definitely you don't want to happen. You know, she's here. This is actually a phosphate standard. So I believe when we shot this video, she needed to run calibrations. which we do every single day, to create a curve. So she's just refilling it up. Something amazing about the discrete analyzer that we have is that it's able to use very small amounts of standards and sample for the analyses, which means we produce a lot less waste, chemical waste. There is another type of auto sampler that exists that's commonly used as well, and that would be the continuous flow analyzer. So that's another common. It works a little different, but it's same methodologies essentially. But we went with this specific, the discrete analyzer, because it does allow you to run multiple types of chemistries at a time. We're not limited to just kind of a few of them, you know, 2 or 4 at a time. So that worked for us. And what she's actually running here, those are the sample cups that you see there. And she put in a calibration standard, or certified reference material. So again, we do a lot of quality control. We want to make sure everything is reading accurately before we actually run the samples. So she placed standard that we bought from a third party, and approved third party, and you know, she's making sure it's reading accurately. And we have this software, and so you can see here it has a robotic arm. And that thing knows exactly what to do. We tell it, say, run this method that we programmed, and you know, it's going to pick up the samples, the reagents, everything that it needs, the buffers to create that reaction. And it makes sure that it cleans itself to avoid any cross-contamination. So that's why it's constantly going back to that wash. Every single time. You can see closer with that water bubbling out, it's cleaning from the inside and the outside. Every single time it goes to pick something different. And essentially this technology is using a colorimetric technique. And so what that is, you're testing the sample solution by measuring the absorbance of a specific wavelength of light. So eventually, you know, on the other side, when it's kind of finished reacting, it'll get transferred to the optical glass cuvette, and it'll read that absorbance. So the colorimetric technique, I'll use ammonia as an example. So it's not easy. I don't think anyone really does it. You can't measure ammonia directly. So what you do is you mix your sample with 2 types of reagents, right? In this case, you know, alkaline phenol and sodium hypochlorite, like bleach, and it reacts with the ammonia that's in the sample to form what's called indophenol blue. So it's like a bright blue color. So now, like, you know, then you can really read this. So that's proportional to the amount of ammonia present. So you're kind of indirectly measuring. Right. So next, we also are able to conduct total suspended analyses, total suspended solids analyses. Now, this is not part of any existing regular program, but we already have the equipment there. And actually just recently, Bill Lucy, our soundkeeper and one of our fisheries biologists, you know, sent me samples to process last week. You know, if there's suspicions of like these discharges, you know, they can confirm it, you know, by sending the sample to me. And then harmful algal blooms. And we came up with this idea, you know, buying the microscope and the camera, because, you know, I mean, most know harmful algal blooms, you see like either like a slimy lime green color or red tide or brown, and you're like, whoa. And you hear about, you know, some of the harmful algal blooms can, you know, unfortunately kill or make dogs sick, humans too. And, you know, we don't want that. So I believe it was years ago, we reported it to New York. But they tend to really focus on like ponds and lakes, more freshwater bodies of water, and not, they weren't able to at the time head out to the harbor that we saw this like slick of like lime green color on the surface, which was, we're pretty sure was, you know, an algal bloom. And they were like, hey, well, can you guys just collect the sample, look at it and confirm for us? You know, we're kind of, you know, I don't know if they were shorthanded or just not able to go out right then and there. But at the time we didn't have, you know, the microscope and camera work to do it. So we're like, you know what, let's have this on hand so that if we get another report, you know, we can really confirm it. And then, you know, for everyone's safety, really put something out or let the state know, and they can really take action with that confirmation. So we have that capability. Also, I was kind of happy to get a microscope in the lab again, since that's where I started looking at diatoms, kind of missed it. So I want to talk about what's next. When we planned our laboratory, we really had the thought of like, what type of future analyses would, like, we need the space to do this. And microbial source tracking is what we want to do next. It's an expansion of our bacteria program. So if you recall, I did mention that the indicator bacteria that we looked at, you know, can come from any warm-blooded animal. So I can't technically say that, you know, the elevated levels of this from the sample is definitely human or sewage. It could have come from geese, dogs, wildlife. You know, and usually there's other indicators, especially if you have consistently over the top levels and you're collecting it from a, you know, outfall or pipe, you know, you could, and there's a smell, you can probably make a great guess that it's might be untreated wastewater. But, you know, to determine the actual source by looking at the DNA, and say, hey, yeah, this is human, or no, this is geese. Then you can take, you know, the towns, the people, you know, we can really take appropriate action depending on, you know, what the source is. So with that, we need a PCR system here, like a little nano drop, which I wish I had a video. It's so cool. I mean, you put the tiniest little drop in there. It's how you quantify how much DNA is in there. It's crazy. I get really excited about the instruments that we get. They're pretty cool, but they do amazing work. They're really important. And we did put a proposal, so we'll see what happens next. Currently, we, you know, occasionally send out our samples to a contract laboratory that can do this, but we would really lower costs and I think fill a need in this area if we could do this ourselves. Not to mention, if we get a PCR system, we can also do what's called eDNA, environmental DNA, and we can partner with our ecological restoration team when they do fish surveys, because they also currently collect water samples to send out for, you know, to see, oh, you know, how much like river herring is based on DNA that they leave behind in the waters. So that's something that we can kind of start doing in-house as well with this system. Next up is coastal acidification, and we really want to make this a tier 3 of unified water study. And so often people will hear about ocean acidification, and there is data currently collected out there in the deeper waters, but again, there's not as much data available regarding, you know, coastal acidification. And ocean acidification mostly gets, you know, absorbs the atmospheric carbon dioxide. That's kind of like the main source. But with in the coastal areas, bays and harbors, there are some additional factors that kind of complicate, you know, what's going on. And, you know, you can see in this image here specifically, you know, it's freshwater flows, urban and agricultural activities really can add to it. It's not just, you know, or increased atmospheric carbon dioxide in this specific area. And then of course, you know, you have, you know, direct nutrient inputs from stormwater, you know, wastewater discharges. So we, you know, not just us, but many are, you know, we have our eyes on monitoring for coastal acidification. So again, this is really much related, all the whole running theme of like, untreated wastewater, nutrients, all of that kind of getting in there and really throwing off the complex balance of biochemical processes that currently occur, and making sure that we make it of good quality for aquatic life, not to, and when you don't hear it impacts shellfish, coral reefs, that carbon dioxide that gets absorbed really messes with that chemical processes and limits the availability of that calcium carbonate that is really important for shells and skeletons. And it throws a lot of other things off. So we, again, we're putting in proposals, looking around to get the lab instruments for dissolved inorganic carbon analyses, organic carbon analyses, and total alkalinity. There's a few other things, but that'll be done out in the field. And another thing that's next is getting ELAP approved. This is a program that it's actually a national program that New York also participates in, and they provide accreditation. It's a whole approval process. It's been long in the making, and I'm happy to say that we submitted our application back in mid-December of 2023, so it's undergoing review. It takes time. I was so happy about that. There's a lot of materials and work behind that, and we have to complete 2 pass-in proficiency tests. So it's basically getting a blind sample and then saying, okay, this is what we got, and we got it right. And we passed our first PT test last month. I was so nervous. I got the result and I just couldn't, it was like, I was driving into work and I was like, I knew an email came, but I couldn't read it because I'm driving. So as soon as I got, me and Lindsay just jumped 100%, we passed with flying colors. So I was really happy about that. Even though I know that we do good work, it's still nerve-wracking, especially when it's your first time. So now we are currently working on our next round of proficiency tests. So now we're processing our next set of blind samples. And then once we submit those, we have an in-person laboratory audit where it can take up to a day, and they'll really kind of see how, we operate the lab. So they'll really get in there, and then we would get our approval. We will. I was going to say I hope so, but we will. I'm determined. It's going to happen. So that should happen, probably April, May or so is what we're looking for this whole process to be completed. And then actions for cleaner water. You can reduce water usage, maintain your sewers, make your voice heard. That has worked so many times where we've had the community hey, what's going on, to their town officials. Eliminate or reduce fertilizer use, keep litter out of the waterways, and pollution watchdog. We'll share that link, but you can go on our website, and if you have suspicions of manhole overflowing or any other sewage pollution, report it to us and we'll look into it. And then you can volunteer. If you go to our website, you can see many different ways of volunteering with us: bacteria monitoring, cleanups, event support, tabling, and restoration. And I'll share a little bit more about bacteria monitoring, since that's kind of our program. So I'm going to really advertise that. But I want to thank our supporters. Due to a very generous gift from the John and Daria Barry Foundation, hence why we named them after them, we were able to have this lab built and get that nutrient analyzer. And then also I want to, shout out to those who also helped us and get to this point with the laboratory. So very meaningful. And then, if you're interested in collecting water quality samples for bacteria analysis, definitely, put your phone to the QR code, or you can just email me and I can send you more information. But yeah, we collect, you see how many samples we collect. We definitely need help. I do remember during the pandemic, we couldn't have volunteers, and I've always appreciated volunteers, but I really appreciated them that year. That was hard, collecting all those samples with just me and 2 others. So yeah, always looking for volunteers. But any questions?
And if anyone has questions, feel free to unmute yourselves or to put it in the chat, and Amanda or I can read them off for you.
Does this mean I was super thorough?
I also just
Okay.
want to add that I will be sending around the recording later this week. And if you think of any questions later on, you can also email us.
Hi, I did have a question. How far up do you do the sampling? Does it just go up to New Haven?
Oh, for bacteria monitoring?
Yeah.
Yeah, we only go up to around Greenwich, Connecticut, and then we do some sampling in Queens around Long Island or Little Neck Bay. And I didn't mention it earlier, that's because bacteria samples have a short holding time. They need to get analyzed, they need to be back in the lab within 6 hours of collection. So there's no wiggle room. So because our laboratory is located in Larchmont, we are limited in terms of distance of how far a sample, can be collected. And also, there are, when you go further into Connecticut as well, there are other existing organizations who we partner with on the Unified Water Study that do bacteria sampling. So we don't want to have to duplicate efforts if there's already another organization kind of doing the work.
Okay. Yeah. No, I was looking up around the Guilford area, and
Yeah.
and I guess they do something around. There's another organization because I checked about volunteering, but I do want to say I graduated with an environmental science degree from Hunter College 47 years ago. And it's great. It's wonderful to see the changes. When I'm looking, we used to always talk about the micro amounts, so we didn't have so much waste. And I'm looking at the lab, and I can appreciate some of the labs I worked in, but it's nice to see a nice clean lab with, small amounts of waste being generated. So very nice. So very nice presentation.
Thank
So
you.
I'm hoping to be able to volunteer with this now that I'm retired. So
yeah, wonderful.
Okay.
I'm happy to hear that. Yeah.
Thanks.
Yeah. I've seen some labs, and actually when I worked in Philadelphia, sometimes I would go into the fume hood. I had to add like nitric acid to digest, my diatom samples, and someone would have, I don't know, like I don't know, it was like maybe food, moldy stuff, like just sitting. I think they would just randomly like play around with testing other things that they could, because there was I don't know, so many labs. So they've been around for probably, I don't know, 70 or so years. And so, you start to do some weird stuff, I think, in these labs. So it was pretty cramped, and it gets kind of messy.
Well, congrats on the certification. I'm sure you'll eventually get it. I know you need it. So
thank you. Yeah.
All right. Oops.
All right. And I was just going to say, with that, we're just about at time. So I wanted to thank everyone for tuning in, and thank you, Elena, for sharing all of that with us today.
Thank you for having me.
And as Amanda mentioned, we will be following up with a recording. Oh, go ahead. Is someone jumping in with a question?
No, I was just going to say thank you.
Oh, thank you for coming. And again, feel free to write down my email. So if you think of any questions later, just shoot me an email. And if you're interested in I didn't go over how to collect these samples, I'm happy to, answer any other questions outside of just, the actual lab analyses.
Okay, and we have a couple other thank yous in the chat. And with that, enjoy the rest of your day. Thanks, everyone.
Bye, everyone.
Thank you.